Hello, and welcome to everyone who's managed to join us for today's live online event. I'm Professor Sasha Hooker, and I chair the Biology, Equality, Diversity and Inclusion Committee. In 2017, we began celebrating the UN International Day of Women and Girls in Science by holding an open lecture to showcase the work of a particular female biologist. Today is our fourth such event. So for everyone who's tuned in, I think we're mostly getting used to these live and recorded Teams events now, but for any newcomers, please type questions into the Q&A that you'll see at the top right of your screen. After the talk's finished, Professor Frank Gunmore, the head of biology and a keen supporter of our equality, diversity and inclusion initiatives, will moderate a question session. Bryn Stewart, our student president for the School of Biology, has created an online form for this event, and you'll get an announcement for this during the talk. It's always useful for us to see who's attended and to receive feedback, so please do try and fill this in for us. And now I want to hand over to the principal of the university, Professor Sally Mapston, to say a few words about this event.
Good afternoon, everybody. Each year, the School of Biology's Equality and Diversity Committee organises a lecture to mark the United Nations International Day of Women and Girls in Science. This series provides us with an opportunity to showcase examples of the most outstanding science being done by women, and it is a real privilege in that context to welcome today's speaker, Professor Cassie Stoddard of the University of Princeton's Department of Ecology and Evolutionary Biology, who will shortly deliver a talk entitled The Extraordinary World of Birds and Their Eggs. Professor Stoddard will be introduced more fully by Professor Susan Healy in just a moment, but I would first like to speak about this day and its relative importance to us as a university, as well as to the higher education sector more broadly. The United Nations General Assembly declared that the 11th of February would be International Day of Women and Girls in Science in 2015, in recognition of the fact that, and I'm quoting the UN here, a significant gender gap has persisted throughout the years at all levels of science, technology, engineering and mathematics, and that even though women have made tremendous progress towards increasing their participation in higher education, they are still underrepresented in these fields. We can see this progress regarding increased participation in our School of Biology at St Andrews. Today, our overall student population is 58% female to 42% male, whereas in the School of Biology, the student ratio is 69% female to 31% male or non-identifying. That's a really overwhelming majority, which has been consistent for the last 5 years, in which we've recorded detailed institutional statistics on gender. Institutionally, despite being majority female at undergraduate and postgraduate taught level, this gender balance shifts in favour of men at postgraduate research level, suggesting that fewer women progress up the academic ladder. The PGR ratio is 46 female to 54% male. But the School of Biology is leading here too in retaining women as postgraduate researchers with a majority female population for the past 5 years. This year, that figure is 64%. This is a crucial measure because whilst we must provide routes for women into STEM subjects, we also want to ensure that they thrive and progress in those areas. And this demonstrates that our School of Biology has a track record of ensuring its students excel on a par and to advanced levels despite their gender. We know that more can be done in other STEM subjects, and we are working to improve participation in those areas, with biology serving as an exemplar. But as the UN's objective tells us, whilst participation has increased, underrepresentation of women in STEM subjects persists at advanced levels, and we see that at St Andrews too. The staff ratio in the School of Biology is roughly equal, 48% women to 52% men. But what we see is a levelling off of the numbers of women at more senior academic levels. At lecturer and senior lecturer level, there is an almost equal gender split. But when you move up the academic ladder, there are 2 women readers compared to 8 men. And at professorial level, women represent under a fifth of professors, 5 women to 22 men. This pattern is broadly replicated across our university and indeed across the national higher education sector. The reasons for these imbalances are complex and they are intergenerational, and they include a historical hangover from when education in STEM was not as hospitable for women as it is now. The answer to realising change is thus to implement strategies which view diversity holistically. No one programme or intervention can fix the problem, but change must instead be generated through many interventions and provisions which support women through their careers from beginning to end. This means that fixing existing imbalances cannot happen instantaneously, and that can be frustrating to come to terms with. But sustained commitment and meaningful interventions have improved the situation for women in academia thus far, and will further change the landscape as we intensify our efforts in this area. This is a real priority for us as an institution, and diverse St Andrews is a key theme in the university for 2018 to 2023, and I lead on that personally. Anybody interested in that work can find more information, of course, on our website, or you are very welcome to contact me. Just one of the things we can do to foster this cultural shift is to provide role models. It is often said that role models are important for women, but female role models are actually important for everybody. Changing our culture involves all of us seeing the value of doing so. This series hosts women whose exemplary careers can inspire both current academics and future generations to excel. Professor Stoddard and her work have to be seen as an example to us all of the immense value that female scientists add. Before Professor Susan Healy introduces Professor Stoddard in greater depth, I do wish to thank the organisers of today's event, Professor Sasha Hooker, Professor Frank Gunn-Moore and Bryn Stewart. As you may already have gathered, our School of Biology is a leader in realising the progression of gender equality at our university and in the sector. It is one of the 3 schools at our university with a Silver Athena Swan Award, and the feedback from Advanced HE on that submission described it as a school in which, and I'm quoting again here, equality and diversity work is clearly embedded rather than being driven by Athena Swan cycles. That is a commendation which we must all strive to emulate. So thank you to everyone for joining us, to our organisers, and most importantly to Professor Cassie Stoddard for the information and the inspiration she will soon share with us all.
Thank you very much for that perspective. Now I would like to introduce the Director of our Centre for Biological Diversity, Professor Sue Healy, who will tell us a little more about today's speaker.
On this Darwin Day, it gives me great pleasure to introduce the 4th of our annual public lectures to mark the United Nations Day of Women and Girls in Science. One of the things we would like to achieve with these lectures is not just a recognition of the fundamental contribution made by women and girls to science, but also to showcase and provide inspiring examples of substantial contributions to the understanding of our natural world. In so doing, we have chosen individuals who inspire not just for the quality and quantity of their contributions, but also, even in the course of their relatively short lecture, make us think about and marvel at that world. Women who inspire us not just by evidence of sheer hard work, but whose passion and enthusiasm for finding out about that world send us off feeling that this day is a better one, and that the pursuit of scientific knowledge is hugely rewarding, exhilarating, and above all fun. This year's lecturer, Cassie Stoddard from Princeton University, is, as you may have noticed, rather younger than our previous speakers, demonstrating that one does not need to be well advanced in age or stage of career to be inspiring or to have made significant contributions. I will leave Cassie to tell you about her science, but she comes to us today from an associate professorship in ecology and evolutionary biology at Princeton via a Marshall Scholarship in 2008, scholarships that are awarded to intellectually distinguished young Americans to study at any university in the United Kingdom. Cassie took hers to Cambridge. Her PhD work there led her to winning the 2012 Zoological Society of London Thomas Henry Huxley Award and Marsh Prize, which is awarded each year to the top zoology thesis in the UK. This award, which followed awards during her undergraduate degree at Yale, has since been followed by awards and prizes across a range of fields in biology, from ornithology, with the Ned K. Johnson Young Investigator Award from the American Ornithological Society for outstanding and promising work by an early career ornithologist, through animal behaviour and the Animal Behaviour Society Outstanding New Investigator Award, to evolution and the Dobzhansky Prize for Outstanding Young Evolutionary Biologists from the Society for the Study of Evolution. Suffice to say that the list is long enough that if I were to itemize each award, Cassie would have no time to deliver her lecture. But it is important to say that her contributions have already been recognised beyond biology, with awards from L'Oreal in 2015, where she was identified as an International Rising Talents Fellow, one of 15, only 15 internationally. In 2018, by a Packard Fellowship, fellowships that are aimed at providing the nation's most promising early career scientists and engineers with flexible funding and the freedom to take risks and explore new frontiers in their fields of study. Now she is a junior fellow in the Harvard Society of Fellows. As the principal has already said, Cassie was due to speak to us last year when history overtook us, but we are delighted that she is happy to deliver her lecture online to us this year. Now we are all the more in need of the colour of her work and the reminders of the wonders of our world being revealed. So ahead of your talk, Cassie, thank you so much for being here today. And now over to you.
Wow, hello everybody. Thank you so much for those very generous introductions. I am really honored to have been invited to participate in this celebration of women and girls in science. And I'm going to pretend that I am there with you all at St. Andrews, particularly because St. Andrews is a special place to me. My grandmother actually spent a semester studying there in the 1930s. Here she is. And it was an experience that she really cherished. I was thrilled to be able to visit St. Andrews once during my PhD in the UK, so I can picture your beautiful campus. It was actually both of my grandmothers who first got me interested in birds, which eventually led me to biology. And I feel very fortunate now to spend my days doing research with an excellent team of students and postdocs. And the work I'm going to share with you today is really the result of our collaborative efforts. In my group, we are interested in the evolution of form and function, especially with respect to animal coloration and morphology. So we tend to look at a scene like this one and we ask, what are the evolutionary processes that have contributed to the tremendously diverse forms we see here? And what are the functions of these remarkable signals and structures? And in my lab, we tend to focus on birds and on their eggs. And we do that for a few reasons. The first, and maybe the most obvious, is that birds are extremely colorful. They can be vibrant and ornate, like the birds you see here. They can also be cryptic and subtle. Their diverse colors are produced in a variety of ways with different pigments or with different nanoscale structures in their feathers. And we know that these colorful feathers are often important visual signals. These signals can function in a range of contexts, from mate choice and signaling to recognition and camouflage. Another reason to study birds is that they have excellent color vision. Birds have 4 color cone types in their retinas, and one of these cone types is ultraviolet sensitive. Now we humans, by contrast, have just 3 color cone types. Birds also have oil droplets in their cones that act as filters, and that gives them really refined color discrimination. And all of this means that many birds will appear even more colorful to each other than they do to us. So hopefully you can see here a male painted bunting. This is one of the most colorful songbirds in North America. And if you use a spectrophotometer to measure his bright green back, you will actually see that this back is not green. It's actually ultraviolet green. So we humans can detect light between 400 nanometers and 700 nanometers. That's our visible range from about blue up to red. But birds can see all of these colors plus ultraviolet in the 300 to 400 nanometer range. And it turns out that many birds reflect ultraviolet light in their plumage. Here are 2 photos I took of museum parrots with a UV sensitive camera. So here's the first image. This is the normal human visible range. And here's a UV image. And the bright white patches in the UV image show that there's actually a lot of UV color on these birds. We just can't see it with our own eyes. Now, even though they're a little less flashy, the eggs of birds are also remarkably diverse in their color and patterning. Egg appearance is often associated with camouflage in different habitats or sexual signaling, but color and pattern also play a crucial role in the evolution of egg mimicry by brood parasites. So if you look very closely, here is a cuckoo egg that has been laid in the nest of a reed warbler. So birds have evolved color not just for communication and for camouflage, but also sometimes for deception. Eggs are noteworthy not just for their color and pattern, but also for their variation in shape and structure. Now, at first, these interspecific differences might seem a bit surprising because all eggs have a very clear function. And that, of course, is to nourish the chick up until the moment that it hatches. But eggshell is actually the ultimate multifunctional material. It has to be tough and weak. It has to provide antimicrobial defense. It has to facilitate gas exchange. It's important in thermoregulation, and it has to provide camouflage or signaling functions. So given all of that, I think it actually makes sense that different species have solved these challenges in different ways. And that makes eggs a really powerful system for investigating form and function. We know that eggs are under extreme selection pressure, but we also see a variety of forms. Eggs are convenient because they're simple, they exist in a straightforward microhabitat, and they don't yet have complex behavior, but they can provide important insights into behavior and evolution. So using birds as the source material, the big questions I want to explore today are what evolutionary and sensory processes shape the diversity of signals and structures in the natural world, and what mechanisms are responsible. To do this, I want to tell 2 different stories. The first is about the visual world of hummingbirds. We've been trying to understand how hummingbirds see color and how this relates to their courtship and their foraging behavior. And then I'll switch gears and explore how egg pattern and shape and structure can shed light on aspects of avian behavior and evolution. So this brings us to the star of the first story. This is the broad-tailed hummingbird, and we believe that hummingbirds are tetrachromatic, like almost all other diurnal birds. That means that they have 4 color cone types in their retinas, sensitive to ultraviolet, short, medium, and long wave light. So in other words, they have a UV cone, a blue cone, a green cone, and a red cone. By contrast, most mammals are dichromatic with just 2 cone types in their eyes, sensitive to short and long wavelengths. We humans are trichromatic with 3 color cone types. These are our blue, green, and red cones. Bees are also trichromatic, but they have a UV cone type instead of a red cone type. It turns out that birds have actually retained the ancient vertebrate color vision system with these 4 different color cone types. Mammals lost 2 of the color cone types early in their evolutionary history, and we humans enjoy our modest color vision only because the old world primates re-evolved a 3rd color cone type. So we humans have this kind of basic color vision system compared to the tetrapod ancestor. But what does it really mean to be tetrachromatic? Well, we know that color vision has to do with the way that the different cone types interact. So it's not just that birds can see all of our colors plus some UV tacked on at the end of the spectrum. In theory, they can see all kinds of combination colors like UV green and UV red. And we think this gives birds an extra dimension of color perception. In humans, the colors we see can be represented in a simple triangular color space in which each vertex represents one of our 3 color cone types. So here we have our shortwave sensitive blue cone type, our medium wave sensitive green cone type, and our long wave sensitive red cone type. And when we see white, all 3 of these cone types are equally stimulated. When we see turquoise, our blue and green cones are equally stimulated. Now, because birds have a 4th color cone type, we have to represent their colors in a tetrahedral color space. And we currently assume that birds can see colors throughout this entire space. And that is a convenient assumption because it gives us a way to measure and analyze bird colors in a manner that's relevant to bird vision. So in other words, we can measure the reflectance properties of the blue crown or the UV green back of this painted bunting. And we can combine this with information about the sensitivities of the 4 bird color cone types. When we do that, we can estimate how a bird's eye would be stimulated. This allows us to map plumage colors in an avian tetrahedral color space. But we still have a pretty limited understanding of how birds actually perceive colors throughout this space. And in fact, psychophysics experiments on color vision in the wild are extremely rare. And so even though in theory, birds might perceive colors throughout this space, that hasn't been well tested. Most work on bird color vision has been done in the lab on a few model species using just a handful of individuals. And those experiments have been extremely important. But we lack a non-human vertebrate system for investigating color perception in the wild. We really need to design experiments that have ecological relevance and allow us to study individual variation in a population. So we took inspiration from some early work on color vision in hummingbirds and bees, and from more recent work on wild hummingbird cognition, including research by Sue Healy and colleagues. And we turned our attention to the broad-tailed hummingbird. So these broad-tailed hummingbirds live for part of the year at our field site. Our field site is in the Colorado Rockies, and we work at the Rocky Mountain Biological Laboratory. So hopefully you're seeing sort of a fly-in now to our field site, which is at about 3,000 meters elevation. The broad-tailed hummingbirds spend their summer breeding here after migrating north from Mexico. This is what the field site looks like most of the year, covered in snow. But by the time we arrive in early June, it looks like this. So with my colleagues, including Ben Hogan, David Inouye, and Harold Eister, who are shown here, we've been training a population of wild hummingbirds to participate in color discrimination tests. And I say training, but actually these birds require very little training because they will visit feeders readily. So these hummingbirds are wonderful and energetic test subjects. And at our field site, we hang up feeders to attract the birds. We eventually take down the feeders and we replace them with our experimental setup. We designed these special bird vision LED light tubes. We call them the tetracolor tubes, which allow us to create many bird visible colors, including those with UV light. So we can make colors like UV green and UV yellow and UV red. Next, we performed experiments that work like this. One color is rewarded with sugar water and the other has just water. After a set amount of time, we swap the positions of the tripods so that birds can't simply use spatial cues to select the rewarded color. We then record whether birds choose the correct rewarded color when they visit the experiment, which they do several times over the course of the day. And after several hours, we can detect whether the population has learned to discriminate the colors or not. So in this case, the birds can easily discriminate red from green when red is the rewarded color, and green from red when green is the rewarded color. And we match these colors so that they'll have equal brightness. We also perform control experiments which show that birds can't distinguish between identical colors when one tube is rewarded and the other is not. And that might be the case if the birds were simply using social or olfactory cues. So we've performed this control experiment now with lots of different colors, and the control works in those cases. So we do believe that color is the key variable of interest that the birds are really paying attention to here. So here's just a brief clip showing the birds participating in one of our experiments. Let's see a bird coming to get a drink at one of the tetracolor tubes. And the experiments I've outlined here, like red versus green, these are pretty simple experiments, but we've been using the same paradigm to perform more complex color discrimination tests. So one of the big questions we've been asking is, can hummingbirds actually perceive colors throughout this entire avian color space? This is a fundamental assumption of the color vision models that we use to estimate color perception in birds and other tetrachromatic animals. And it relies on the idea that birds can see non-spectral colors, which are not in the rainbow. But this idea has not been tested systematically. So what are non-spectral colors? It might help first to focus on spectral colors. So the spectral colors are those that are in the rainbow, and they're generally represented on the three sides of the tetrahedron that I'm showing here in color. So you can use monochromatic lasers, and researchers have, and they've shown that birds can see and discriminate these spectral colors, which stimulate either one or two color cone types. So in other words, here's pure UV color. This would stimulate just the UV cone. Here we have a color that would stimulate the UV and blue cones. This color would stimulate just the blue cone, and so on. Sort of move around the edges of the tetrahedral color space. But what about more unusual combination colors? The kinds of colors that would allow birds to perceive colors throughout this entire space. These are colors that fall on the three other axes, those shown here with dotted lines, the UV red, UV green, and red blue or purple axes. So we have UV green colors, which would stimulate the UV cone and the green cone, but not the blue and the red cones. We have UV red colors that would stimulate the UV cone and the red cone, but not the blue and green cones. And we also have purple, which would simultaneously stimulate the blue and the red cone types. So these non-spectral colors are those that would stimulate pairs of cones that are sensitive to widely separated bands of the spectrum. They're called non-spectral because they're not found in the rainbow. And we don't know much about whether and how birds can see these colors. And we've already seen today one non-spectral color, the UV green back of the painted bunting. But would a bird really see this as UV green, as something distinct from pure UV and distinct from pure green? That is the question. Humans have only one major type of non-spectral color, that's purple. This arises when our blue and our red cones are stimulated, but not our green cones. Purple isn't actually in the rainbow. It's a special color we see because we have trichromatic color vision. And birds have several additional possible non-spectral colors, as we have seen. So we've now conducted a series of experiments to determine whether hummingbirds can discriminate pairs of colors on the three non-spectral axes, here, here, and here. And our results show that hummingbirds can discriminate these colors with high probability. So here's a summary of the results of 19 experiments, which show that the birds were able to distinguish between pairs of non-spectral colors that more or less fall along the dotted non-spectral axes that you can see here on the left. So overall, this suggests that hummingbirds do see a huge range of combination non-spectral colors that we humans can't imagine. This probably is true not just for hummingbirds, but for other birds and other tetrachromats more generally. So these results provide strong support for the tetrahedral color space model. We of course don't yet know which neurons in the bird visual pathway might be responsible for processing these colors, so there's still much more work to be done. But we do know that these non-spectral colors are prevalent in the natural environments of birds. So here are three reflectance spectra showing UV red, UV green, and purple plant and plumage colors. We took this one step further and we estimated that these non-spectral colors are much more common for birds and other tetrachromats than we realized. Since we humans have just one major non-spectral color, which is purple, when we analyzed 1,000 plumage colors, we could estimate that nearly a third of these would be seen by birds as non-spectral. Now, I don't want to suggest that non-spectral colors are more important to birds than any other color or that they're somehow perceived in an extraordinary way. You know, these colors aren't glowing or popping out. These are just a consequence of having a tetrachromatic visual system. So the message here is that these colors exist in nature and birds can and do perceive them. Overall, we're using this experimental approach to test fundamental questions about avian color vision, but we're also interested in asking specific questions about how color perception relates to hummingbird behavioral ecology. And to do that, we have to understand what colors matter to hummingbirds in their daily lives. So with this in mind, we've been investigating the role of color in their courtship behavior and in their foraging behavior. The male broadtails perform these spectacular courtship dives in which they fly about 30 meters up into the air and then they plunge to the ground just over the head of the female and then fly back up again. It's very dramatic and they do this repeatedly. At the bottom of the dive, the birds will snap their tail feathers together to create a loud buzz, and they also show off their iridescent throat feathers. We know from work by Chris Clark and his colleagues that this loud buzz is produced by the tail feathers. So with my postdoc, Ben Hogan, we wanted to know, how does color interact with motion and speed and sound in this dive display? How does the female perceive these aspects of the dive, and how synchronized are these different elements? And we were especially excited to think about iridescent color, since the male broadtails have really striking iridescent feathers, which change in appearance quite dramatically as a function of viewing angle. It's been relatively easy to study visual signals that are static, but of course real animals move, and the dive of the hummingbird represents a signal that is complex and dynamic in both space and time. In the field, we recorded videos of dives and we used tracking software to track the path of these dives. So here's a male that performed 2 sequential dives. From the tracked dives, we could then calculate some flight performance statistics. So for example, we can estimate that males are going about 25 meters per second at the bottom of the dive, which is really fast. We also analyzed the sounds that males make in the dive. So here are sonograms from 5 different dives, and they all show the same pattern. As the male is going up, he makes a trill produced by the wings. At the very bottom of the dive, the male will snap his tail feathers together to make that buzz that I mentioned. Using this acoustic information, we could start to map these sounds onto the trajectory of the dives to see how everything times up. Based on the male's speed and his trajectory, we estimated that the female will perceive a shift in the frequency of the male's sounds as he approaches and departs due to the Doppler effect. So it's really dynamic reception of this sound. The final component here was color. We used a multi-angle UV and human visible photography setup to capture iridescence of plumage using museum specimens. We designed a 3D printed stage for hummingbirds, and we attached specimens to it, and then we rotated and imaged these specimens at fixed angles. So here's a video that highlights the degree of that color change. And we did this work at the American Museum of Natural History in New York City, where Paul Sweet, who is the ornithology collections manager, has really been a critical facilitator of this work. Museums are very important for the kind of work we do. We then could use a model of avian color vision to estimate how the female's color cones would be stimulated by the male's iridescent throat. This showed us that as the male dives toward the female, he'll first appear bright red, but as he reaches the very bottom of the dive, his throat will appear to change quickly from red to a blackish green. So putting this all together, for each dive, we could investigate the timing of these 3 key features. Here's a simulation from one dive, and it shows that this male is delivering a sensory explosion right at the base of the dive. So at this moment, the perceived color of the iridescent throat changes from red to black, the male's making his tail-generated buzz, and he's reaching his top speed. When we analyzed all of the dives together, we found that this was a general feature. Males tend to reach their top speed at almost exactly the same time that they snap their tail feathers together and show off their iridescent throats. So in other words, all of the action is happening at once in this concentrated 300 millisecond window, which is literally a blink of an eye. So now we have a better understanding of how these remarkable displays unfold. But the question remains, how is the multimodality really functioning? Are these different sensory components intended to show off the male's quality or flight performance, or do they provide backup so that a male can still show off his color in a noisy environment or show off his sound on a cloudy day? Or do these signals work together to increase detection or memory? Or is somehow the combination and synchronization of these signals just super sexy to females? These, of course, are all possibilities, and we'll have to do more work to figure out which of these explanations might be right. So turning now to foraging behavior, at our field site, every week brings a new wave of wildflowers. Surprisingly, we actually don't have a very good understanding of which floral species the hummingbirds visit and with what frequency. So to understand how hummingbirds use color, we really have to figure out which flowers they visit. This is especially important in terms of climate change because at our field site, many of these flowers are blooming earlier than they used to, which could cause problems for the hummingbirds if they don't adjust their behavior. My research team has been setting up time-lapse cameras at flowers in an effort to catch hummingbirds in the act of foraging. So here's one of my students, Alice, putting a camera out in the field, and we've so far collected about 30,000 flower hours of footage, and we're now processing these images with a machine learning algorithm. So here are just a few snapshots of birds caught in the act of foraging. Ultimately, we hope this will give us a more detailed quantitative picture of hummingbird foraging behavior. And we're just starting to analyze these hummingbird visitation patterns. Overall, we hope this should provide some detail about how hummingbirds actually are using floral resources and how this might change through time, through a summer or across years. So just to recap, hummingbirds could be an excellent system for investigating color vision in the wild. We can start to understand how color is used by these hummingbirds in the context of courtship and foraging behavior. We found that hummingbird dives are highly synchronized. And of course, real animals move. And so one of the opportunities for our field moving forward is that we can tackle these more complex multimodal displays in which motion and orientation are really important. So now I want to switch gears from color and communication to pattern and deception. Here is my favorite brood parasitic bird. This is the common cuckoo, which will be familiar to many of you. It breeds across Europe and Asia. And I started studying the notorious brood parasitic cuckoo with my colleagues when I was a PhD student, and I've been hooked ever since. They're really fascinating birds. Common cuckoos don't build their own nests, of course, and they don't raise their own young. So here's a cuckoo egg that's been laid in the nest of a reed warbler, and you can see that the cuckoo egg is a little larger than the other eggs, but otherwise it's an excellent look-alike. If the reed warbler parent fails to identify and reject the imposter egg, the cuckoo chick will hatch first and it will throw the reed warbler's own eggs overboard. Then the cuckoo chick grows to a monster baby, and here you can see it being fed by its foster parent. The fact that common cuckoos can mimic the eggs of their target hosts has been well described. And they have to do this because many of the host species, whose eggs you can see here on the bottom row, have evolved the ability to detect and reject parasitic eggs. And this leads to more and more impressive mimicry on the part of the cuckoo. However, we're still learning about how hosts do this, how they identify and reject odd eggs. What cues, and specifically pattern cues, do these birds use to detect and recognize parasitic eggs? I've been working with my collaborators Ben Hogan, Martin Stevens, and Claire Spottiswoode to try to figure this out. We've been using the African brood parasite, the cuckoo finch, which lays eggs in the nest of small birds called Prinias. So no relation to the common cuckoo, but the cuckoo finch is behaving in a very similar way. So here you can see the cuckoo finch eggs in the center ring and the Prinia eggs in the outer ring. Claire and Martin performed egg rejection tests by pretending to be cuckoo finches themselves. First, they swapped eggs around nests to simulate parasitism, and then they checked whether the host birds, the Prinia, rejected the foreign egg or whether they accepted it. From this, they could determine which features were the most important for triggering rejection events. And so here's a Prinia warbler actually rejecting one of the odd eggs. If you are a Prinia host, the cost of making a mistake at the egg stage is very high because soon the undetected egg will hatch. And here you see a cuckoo finch chick. It begs so aggressively after it hatches that it tends to outcompete the Prinia's own chicks, which will eventually die. So at this high stakes moment, which occurs when the Prinia host is staring down at its eggs, it's trying to decide whether or not to toss out this weird looking cuckoo finch egg, what visual cues does the host use to make this assessment? Is it differences in color? Is it differences in simple pattern features like marking size? Well, Claire and Martin found that the Prinia hosts pay attention to all of these things. They pay attention to color differences and differences in simple pattern features. And altogether, the differences in color and pattern between the host and the parasite eggs predicted about 32% of rejection behavior. But truly what kept me up at night was the idea that maybe Prinia hosts are also paying attention to more complex features. So maybe a Prinia host learns that it has a marking like this one that, if you squint, sort of resembles a slug. Maybe it's using that feature to learn about its own eggs. So we reanalyzed the data, this time using a software tool we developed that's kind of like face recognition for eggs. This tool is called Nature Pattern Match, and it can pick up distinctive, complex pattern features. We then compared these features between the host and the parasite eggs, and we asked whether these differences also helped predict rejection behavior. And the answer was yes. We found that birds do pay attention to these more complex features. When comparing eggs, the Prinia hosts care about differences in color and differences in simple pattern features and more complex pattern features. So our new model explained about 37% of variance in rejection, up from 32%. Now this might seem like a small jump, and it is, but it suggests that even after you control for differences in color and differences in these simple pattern features, these more complex, distinctive features still matter to birds. And we hypothesized that these complex features might be especially informative when the host has a really tough choice to make, like when the color and simple pattern features are pretty similar between the host and the parasitic eggs. So to recap, the tools have never been better for quantifying animal patterns, but we need to test which pattern metrics really influence the behavior of birds. We found that in host birds trying to spot a foreign egg, simple and more complex pattern features influence egg rejection. So clearly the bird world is awash with these vibrantly colored and densely speckled signals that can be helpful for communication on the one end and deception on the other. But what can we learn about behavior and evolution by looking at morphology, like the shape and the structure of eggs? To investigate egg shape, we assembled a team of mathematicians and physicists and computer scientists and biologists. Our team included Catherine Sheard, who you can see here, who was a postdoc at St. Andrews at the time. And we asked, why do bird eggs come in such a variety of shapes? How do they get these shapes? Some eggs, like those of owls, tend to be spherical, like golf balls. Hummingbirds lay eggs like little tic tacs. These are perfect ellipses. And many seabirds and shorebirds lay pointy eggs. We were fortunate to have access to the digitized egg collections of Berkeley's Museum of Vertebrate Zoology. And we wrote a computer program called the Eggstractor to select eggs from photographs to fit a mathematical curve to each egg's outline. So from this curve, we determined the egg's asymmetry, how pointy it was, and its ellipticity, or elongation. Ultimately, we were able to quantify the shapes of almost 50,000 eggs from 1,400 species, representing all of the major extant bird orders plus 2 extinct orders. And when we mapped the egg shapes in morphospace, we could see considerable variation not just among the different species, but also among the different bird orders. And you can see some of those orders here. We also proposed a simple biophysical model of processes that happen inside a bird's oviduct. This is the stretchy tube where eggs form. And we suggested that it's not the shell that fixes egg shape, but it's this stretchy membrane. By adjusting the membrane properties and varying the pressure that's applied across the egg, we could generate lots of different egg shapes using this model. And in fact, these eggs whose shapes we could generate included all the egg shapes we had so far observed in nature. We then examined how egg shape changed as birds diversified, and the takeaway here is that there was no one moment when eggs dramatically became pointy or elliptical or round. There have been many dynamic parallel changes in egg shape in these different lineages. Having this bird phylogeny also allowed us to look for associations between egg shape and life history and ecological correlates. And we found that more elliptical eggs and more asymmetric pointy eggs are correlated with phylogeny, with adult body mass, and flight ability. We found that birds that are stronger flyers tend to lay longer, pointier eggs. So why might there be a correlation between flight and egg shape? Well, we don't suggest that eggs have an aerodynamic shape or that egg shape changes when a bird is in flight. Instead, we suspect that wing shape is correlated with other morphological features that could influence egg shape. So for example, as birds' bodies become more streamlined for flight, this might have resulted in smaller abdominal cavities and more compressed, squished organs. One way for birds to still pack a lot of volume into their eggs while maintaining these more streamlined body plans could be to lay more elliptical or asymmetric eggs. We also found that this correlation does not hold in all groups of birds. So in shorebirds, for example, flight behavior did not predict egg shape, but chick development did. So the point here is that the rules of egg shape don't apply to every group, and other selective forces can certainly be at play. Now, thinking about the diversity of egg shape got me really curious about eggshell structure. And so in the last 2 minutes, I'm going to tell you about some work we're doing on my favorite flightless bird. This is the emu. With my colleagues, I'm trying to understand how eggshell structure relates to its dual biomechanical function, which is to prevent breakage from the incubating adult, but ultimately to allow a chick to hatch. We've been studying these beautiful green eggs of emus. These are large flightless birds from Australia. Males actually incubate the eggs in this species, and they'll lose a lot of their body mass while doing so, in part so the egg won't break too soon. So here's a closer look of the eggshell and a microscopy image in cross-section, and this allows us to really see the variation in the shell's ultrastructure as you move from the inner membranes all the way to the outside of the shell. And I'll just highlight one result for now. We've been using a technique called nano-indentation to really figure out how these shells crack. And we found that cracks in the outer region of the shells are deflected sideways, preventing them from penetrating through the shell. But when we look at the inner part of the shell, we find that cracks are easy to propagate vertically at the boundaries of individual crystals. And this might help initiate cracking when the chick hatches. So in other words, what we're finding is some built-in weaknesses that might promote hatching, but also some features that prevent the egg from shattering too soon. So altogether, we're finding evidence that supports the idea that the shell acts to prevent external damage and facilitate chick hatching, all while mediating water loss and gas exchange. So we're continuing to explore how eggshell, which is actually nature's fastest forming biomineral, it's a really cool material, is designed to fulfill these multiple competing demands. So to sum up, I've told you a few stories about color and pattern and shape and structure in the avian world. And the punchline is that birds can provide a powerful window into the evolution of form and function in nature. And particularly when we take an interdisciplinary and collaborative perspective, we should be well equipped to keep learning about their world. And so with that, I want to thank all of you for tuning in, and I want to thank my many colleagues and institutes and funding groups that have made this work possible. So thank you all so much.
Fantastic. That's the first thing I'm going to say straight away. Now, if you have some questions, we do have a little bit of time for some questions. If you look at the top right, there is a box with like squares and it's got a question mark in it. If you sort of type in your question in there, I'll be able to see it, and then I will sort of talk to Cassie and relay the question for you. I guess, first of all, whilst people are typing away, and I can see some people I think are typing away, first of all, I should just say thank you. That was absolutely stunning, really is. I mean, it's times like this, as a humble biochemist, I sometimes feel I did the wrong biology. I mean, to be able to go out into the field and actually, you know, discover completely and utterly new things, I always think is wonderful. So thank you very much indeed, very inspiring. I do actually have a question myself. I do quite a bit of work actually in the biophotonics world, but one of the things I sort of noticed, you sort of said, I think very nicely, that evolutionary, we had sort of like, we were tetrachromatic, and then over time you can lose it, and then you come to us poor sort of mortals with only sort of like 3 sort of possibilities. So it's quite possible that you can lose sort of like one of these sort of windows of vision. Now, it's in the case of birds, are they all tetrachromatic as much as you know? Because one of the things I could see is like, you know, when you had the picture of the what was it, the cuckoo eggs, you know, one was completely different sort of color. So as far as you know, do you think all birds are tetrachromatic, or because some of them are not brightly colored or need brightly colored things, whatever? I guess that's my question.
That's a really great question. Are all birds tetrachromatic? You know, why do we see some weird color differences in the cuckoo eggs, and what could this tell us about bird vision? So far, we do think that diurnal birds are tetrachromatic. There are some discoveries showing that nocturnal birds may have lost some of the color cone types, but to the extent that that it's been explored, and I should say that, you know, there are 10,000 plus species of birds, and we've still only really looked in detail at a handful of these. So I will not go on record as saying all birds are tetrachromatic, but that does seem to be a common feature of birds. But one difference I didn't mention is that across birds, from the tiniest hummingbird up to a large ostrich, the long wave sensitive, the medium wave sensitive, and the short wave sensitive cones, these are the red, the green, and the blue cones, are all pretty similar in their peak sensitivity to certain wavelengths. But the fourth color cone type, the ultraviolet sensitive cone type, comes in two flavors. One is the true ultraviolet sensitive cone, which has peak sensitivity around 365 nanometers. And then the other is the violet sensitive cone, which has peak sensitivity around 405 nanometers. It's still UV sensitive, but birds have one or the other. And so many people are interested in trying to understand, why do some birds have the UVS, the ultraviolet sensitive type, and why do other birds have the violet sensitive type? It looks like the violet sensitive type is ancestral and the UV type evolved multiple times, as many as 15 times in different bird lineages. And there's no rhyme or reason to which lineages have evolved with UV type so far. So this is a kind of burning mystery we're still trying to solve, is why do we get these different variations in tetrachromacy even across birds? So there's a lot to still keep us busy, but that's an excellent question. Thank you.
Thank you very much. So we do have a question here from somebody called Dr. Anonymous. Do birds that swim underwater and therefore have to have streamlined bodies also have elongated pointy eggs?
Thank you very much for that question. This is something we wondered about when we were doing the egg-shaped study. And it turns out penguins, wonderful swimmers with streamlined bodies for swimming, do have pointy asymmetric eggs. So we speculated in our paper that the same kinds of selective forces shaping the body plans of flying birds may be at play in birds that swim as well. And we would love to do a more involved study of just that, to look at the strong swimming birds to see what is shaping their eggs. Thank you.
We have a question from Mike Ritchie. Is anything known about the inheritance of egg color and shape in cuckoos? I've always thought this must be a fascinating and likely a co-adapted gene complex. Showing off technical skills here, Mike. Yeah.
That's also a fantastic question. We do know something about egg color genes, but not nearly enough. So there has been work to, so the hypothesis is that in order for female cuckoos to pass on the egg color and pattern that they have to their offspring, they should have the genes for egg color residing on the female specific sex chromosome. So birds are reversed than mammals. In birds, the male sex chromosomes are ZZ and the female sex chromosomes are ZW. So in theory, if all of the genes for egg color and pattern are on the female W chromosome, that female cuckoo can mate with any old male cuckoo and still guarantee that her daughter will lay the same egg type that she herself laid. This has been something that people in our field have been chasing for decades, and there is some evidence coming out of other research teams is supporting the idea that there must be this kind of matrilineal genetic passing on of the genes, but which genes are involved and their location on the W chromosome remains completely enigmatic. So in my top 5 things I wish I could find out, and I'm not currently working on the genetics of egg color, that would be one of them. It's just a burning question.
Okay, we have another question from a Dr. Anonymous. My project is framed around multimodal signals and how their use may help / hurt species reactions to anthropologic change, and was wondering if this is a topic you had thought on in regards to the birds of multimedia signaling. I have to comment, some punctuation would have helped that question.
I think I got it. It was very well translated. Yes, this is something I think about a lot. I teach a sensory ecology course at Princeton, and it's something very much on the minds of my students, is how are we humans disrupting the sensory worlds that these animals inhabit, and how can we take steps toward preventing that? So in the context of multimodal signaling, it becomes, I think, particularly important to think about what new pressures these animals might be under. And one prediction could be that more multimodality will emerge as animals are forced to have these sort of backup mechanisms, which I mentioned in the hummingbird. So one benefit of a multimodal signal is that an iridescent bird can still make a nice sound on a cloudy day, or they can, show off their color if there's a lot of noise. So they can have more than one way of communicating. And I think it'll be very interesting to see if we see shifts in multimodal signaling in the animal world. I mean, certainly in single sensory domains, we're seeing this already. There's a study that came out last year showing that sparrows in San Francisco have changed the way they're singing because of reduced traffic due to everyone being in lockdown. So I think we're going to see much more of this. It sounds like a fascinating topic that you're working on, and hope I get to hear more at some point.
Okay, we have another question here from Prabs. I think you actually touched on this in part of my sort of question. Do night birds have different chromal vision?
Yes, so this would also make my top 5 list, I have to say, because the hummingbirds are actually nested in a group that contains some of these nocturnal birds, like nightjars. So ancestrally, the hummingbirds' ancestors were these cryptic nocturnal birds, and they emerged with their sister clade, the swifts, from this particular group. And so what that has meant for the evolution of color vision, particularly these opsin genes that are responsible for the color-sensitive pigments in the retinas of these birds, is something we do not know. But one can imagine that there have been certain bottlenecks that might make hummingbirds particularly weird if they've had this nocturnal ancestry. So this is something we actually are working on and are trying to learn more about, because it has implications for how we interpret the color vision behavior of our hummingbirds.
Another question. So yes, back to Dr. Anonymous, who seems to be very busy with her questions. Can you say a bit more about egg shape and how this affects mode of incubation? It has been suggested that the pointy shape of modern bird eggs might have allowed reproduction in a wider range of environments, thus contributing to bird radiation.
Yes, that's a wonderful question about egg shape in the context of incubation. And there are many hypotheses about egg shape variation, and incubation behavior is certainly one of them. We know that across birds, the places they inhabit, from rocky cliff ledges to sort of cut nests, are very different and could introduce different selective factors for egg shape in particular species or particular bird families. In our broad scale comparative analysis, we collected data on the nest locations, or the nest height, and nest type. So are they nesting on ledges? Are they nesting in cups? And we included that as a potential predictor of egg shape variation. And when we looked across all 1,400 species of birds, we didn't find that nest type or nest location was as strong a predictor of egg shape variation as phylogenetic signal, where you are in the family tree, body size, and flight behavior. But when we zoom in on particular groups, there we did sometimes detect effects of incubation behavior and nest type and nest height on egg shape. And so there's definitely much more to learn, and other groups are exploring this. When you zoom in on particular lineages and particular branches, you may find that the selective forces influencing egg shape are not the same as those that you detect at the broad macroevolutionary scale. So this is something we're also hoping to follow up on. We want to understand more what the rules are, and it may well be that when you zoom in on some groups, particularly with extreme incubation behaviors and nest locations, you do see an increased effect of incubation behavior on egg shape. That's something we're working on.
Okay. I should say we're over the time, but I don't care. We're going to keep going, if that's all right, Colin. This is much more interesting than my next meeting, tell you. So anyway, so for the common cuckoo, I know that they have multiple species that they choose to parasitize, and some of the hosts are much more discerning than others. Spelling mistake there. Has it been shown that cuckoos can alter their egg color to suit less discerning host species?
Yes, another favorite topic of mine. So as you can see on that one slide, I showed some of the eggs of the different hosts in the UK. I really focused on UK hosts during my PhD, and some of the hosts are more discerning than others. So in those pairings, the discerning hosts seem to go hand in hand with cuckoos with the best egg color and pattern mimicry. And that makes sense. If a host has been really good over thousands of years at rejecting odd eggs, the cuckoo females that parasitize that host have to be really good at evolving egg colors and patterns that match. But other hosts, like the poor dunnock, the dunnock lays a blue egg, and the cuckoo that parasitizes it just lays this kind of speckled brown egg. And the dunnocks are not discerning. They'll sit on almost anything that you put in the nest. So this does beg the question, what is going on with these birds, and are they at different stages of this co-evolutionary arms race that means that we see these kind of puzzling differences in color and patterning that nonetheless still ensure the success of the cuckoo if the dunnock is powerless to reject most of those eggs. The question that you asked is a very interesting one. Can cuckoos actually change their egg color on the fly to suit a particular host? And no, these cuckoos are not, more or less no. They're not chameleons. They don't lay, female cuckoo doesn't lay a blue egg one day and a green speckled egg the next day. They exist in these genetically distinct host races. So if you're a female cuckoo that lays an olive colored egg, you always try to get your egg into the nest of a great reed warbler. And if you lay kind of a pale blue egg with brown spots, you always try to get your egg in the nest of a brambling. However, there have been some studies, not of my own, but by colleagues of mine, suggesting that cuckoos may be selected within their particular target host species so that they lay an egg that is particularly well matched to a specific nest of host eggs. And this is a really intriguing idea that cuckoos could know enough about what their own egg looks like to be choosy when they're trying to figure out which reed warbler nest they want to lay in. And so more work needs to be done here. There's so many questions about really the behavioral ecology and cognition of cuckoos and hosts that have not yet been explored, despite decades of really fascinating work on cuckoos. So I think there's much more to be learned there.
Okay, this message seemed to have lost our feed going out. Is that right, Colin? So we're back. Okay, so we're back on. So I believe there was a bit of a cutout there. So I'm afraid somebody's question may have not got answered. So great. But anyway, we've got another question here from, and it seems, Cassie, you've got a fan here from Manon. Thank you so much for this exciting talk. I have a potentially weird question. They're always the best ones. Do you think we will find one day a bird species seeing yet another color, and what would it be like?
Oh, Again, the theme of knowing, that's a super question. What could that color look like? Could there be more cone types? I certainly wouldn't rule it out. I'd say never. I mean, we've tended to see these 4 color cone types, but I think the most likely scenario could be duplication of one of the existing cone types to flesh out a little more. Seems to be an issue on my end. Should I keep talking, Frank?
No, I keep talking. Yes, keep going, Cassie.
I will. So it could be that with these 4 color cone types, there's going to be a duplication event that allows some birds to have even better color discrimination within this range. Now, I can't imagine really that this would happen, but I would love to find a bird that had sensitivity in the infrared range. So we haven't talked about infrared, we focused on ultraviolet. There's no evidence that birds can see beyond 700 nanometers, but you never know. You could see something weird like that. So I really hope I hope people will keep exploring this and looking at the diversity of birds to learn more about whether there could be other cones out there.
Okay, I think we may sort of call it to an end there. The reason there may be some technical issues. You may have just heard Cassie give an answer, but just had a look at me and thought amazing that I was like miming all of Cassie's words there without moving my lips. So I think we're having some sort of technical issues. I'm just want to say, you know, once again, Cassie, that was absolutely fantastic. Thank you so much. That was really good. I should say we do actually have a gift for you, and when we can, we will get it to you. It's a bit of a problem because of COVID issues, but for everybody else, you will have seen Bryn very kindly put up a message earlier on, an announcement about filling in the survey. That would be very welcome indeed if you could please feed that fill that in and feed that back. And I think I will wrap up by just sort of saying thank you for everyone for turning up. Thank you, I should say, to Sasha, Sue and Bryn for organising this, and somebody that nobody's seen apart from us once speaking, Colin, who's actually been trying to organise all of the technical aspects. The last word goes to Cassie. That was truly inspirational. Thank you very much. And I hope to see you all again at our next biology talk. Thank you and goodbye.